Hello everyone. I will try to be fast, 3 minutes. So my work is the functional effects of antibody from Chagas disease patients on the electrophysiology of cardiomyocyte divided from human-induced pluripotent stem cells. So Chagas disease is an infectious disease caused by the trypanosoma cruzi that can lead to cardiac dysfunctions, affect the heart, leading to heart failure and also arrhythmias. So those arrhythmic events as ventricular and atrials lead to a high rate of sudden death in Chagas patients. So the reasons that they have a lot of arrhythmias is unknown. So one hypothesis like there is a molecular mimicry between the protein of the trypanosoma cruzi that also can react against like functional epitopes of receptor as beta 1 and muscarinic in cardiomyocytes. So this theory is already like being tested in animal models when we see that these autoantibodies can react and activate these both receptors, leading to change the ventricular repolarizations and change the QT interval and induce arrhythmias. But none of these effects have been tested before in human cardiac cells. So that was the aim of my work. So for that, firstly, we like choosing selected patients. So at the moment, I have like 100 patients. And then after like selection to patients, I have to cross some medical data. So what is the symptoms of these patients? So I collect all data. So today I will present only 3 of my patients. So I collect all these data and later I compare to everything that I see actually in a lab. So the first thing, I need a cardiac cell, human cell, right? So I use like iPS cells from a health donor. So if someone has any disease, a health donor, and then I generate these iPS cells, which I already characterize is already public data test, and then I differentiate into cardiomyocytes. So first thing that I check is how is the expression of troponin. So all my differentiations, normally the started with like 60 until 99% of expression of troponin. And then I did the characterizations of the expressions of the principal ion channels and also some proteins like sarcomeric protein like troponin 10, myosin, and also connexin 43. So after that, I need to do another characterization. So now looking into electrical activity of the cells. So I normally do like the micro, sorry, I'm nervous. So I do the micro electrode when I just have a pool of cells and then a reference electrode in the bath. And then I can test way like recording the actual potential. So the first thing that I did was like T cells actually can evoke one better dynamical response or not. So I use like isoproterenol, and then I saw that T cells can like increase the frequency and I have no change in the direction of the actual potential. So after that, I use like the sera of the patients. So he like 3 different patients, and then I see when I perform the seras in a thyroid solution, I see the increase of the frequency. So you can see the all day increase the frequency. So, and these also change the way that they alter the change of the repolarization. So the patients 1 and 301, they decrease the time for repolarizations after 50 and 90%. And the 82, he do the opposite. He increased the time. So another data is that I could not show here, but we see like these patients also induce an arrhythmias event. So he's like 50% of the plates that are analyzed. Here, 70%, he also 50%. So, concluding, the beta-adenosine antibody 182 3001 had a chronotropic effect and influenced the cardiac repolarizations. So they induce arrhythmia events, premature despolarization, and ectopic beat characterized of ventricular electrical disturbance. So this data is a really new. We never saw nothing like this in the literature, and they highlight the need of study that possibility like understand the signalization of the pathway and maybe can be modulated by these antibodies and how these antibodies can actually generate or just work in the genesis of the ventricular arrhythmias. So that's my group of work, and thank everyone for watching.
Thank you very much. And thank you very much for the opportunity to present our research here. My talk will be about electrophysiological screening of potential drug candidates at the Tetrahydrocannabidiol californica nicotinic acetylcholine receptor to counteract organophosphate poisoning. As you all can see, I'm here with my team from the Bundeswehr Institute of Pharmacology and Toxicology, and we are really interested in single-channel measurement to detect potential nicotinic acetylcholine receptor resensitizers. As some or all of you may know, the nicotinic acetylcholine receptor, we are especially interested in the muscle type. It's a pentameric ligand-gated ion channel of the cis-loop receptor family, as well as, for example, the GABA receptor. And as for the GABA receptor, there are several allosteric modulators known, but right now just for the alpha-7 nicotinic acetylcholine receptor and not for the muscle type receptor. Our aim is to counteract dysfunction caused by organophosphorus compounds. Also, as you may know, organophosphorus compounds inhibit the acetylcholine esterase, leading to a cholinergic crisis, leading to a rise of the endogenous ligand acetylcholine in the synaptic cleft, and therefore addressing the orthosteric binding sites at the nicotinic acetylcholine receptor, leading from an open state to closed state, and after that, if several other binding sites are also addressed, to a desensitized state where no more opening of the receptor is possible and no signal can be transmitted into the cells at the neuromuscular end plate. And therefore, we try to insert the nicotinic acetylcholine receptors into biomimetic membranes using the Nanion system, the Orbit TC, and using black lipid membrane electrophysiology to characterize our potential resensitizers. We had some trouble doing this, to be honest, and the problem with the nicotinic acetylcholine receptor is that it's a really large protein. It's about 280 to 300 kilodalton, and therefore it's really difficult to handle, but we got it in the end. And the secret in this was using proteoliposomes as a transfer medium into our black lipid membranes, using cholesterol, phosphatidylcholine, and phosphatidine phosphate at a molecular weight ratio of 8 to 2 to 5, and mixing these up, drying this using a rotary evaporator, adding additional buffer and diethyl ether, and sonicating it to get a lipid gel, adding more of our phosphate buffer, 10 millimolar, and then drying it again to remove the organic solvent. And after that, we did some manual extrusion through a polycarbonate membrane of about 0.4 nanometer size, getting small unilamellar vesicles, adding what you see here, TC plasma membrane, which means Torbata californica plasma membrane derived from differential centrifugation, which was done using the native electroplex organs of the animal, and adding non-ionic detergents. We tried some ionic detergents before. We tried some cholesterol derivatives, but they don't prove very successful. For us, it was now a non-ionic detergent, which gave us the turn. We proved the formation of liposomes, first the solvations of liposomes, and then the formation of proteoliposomes using absorbance measurement at 300 nanometers. And after adding biobeads SM2, we were able to remove the detergents and transfer these onto our measurement platform. So for last, and most important, our results. What you see first is spontaneous open events of the nicotinic acetylcholine receptor after adding about 100 micromolar of its agonist, or better to say a derivative of the agonist. We use carbamolcholine, not acetylcholine, due to the high hydrolysis rate, and we see spontaneous open events. So to simulate desensitized state or the presence of an organophosphate as no acetylcholine esterases there to be inhibited, we added more agonists. We added 300 micromolar of carbamolcholine, and we see spontaneous open events ceased. And after the adding of a potential resensitizer we had in our eye, we discussed for several years, as other research groups too, we added 10 micromolar of resensitizer MB327, and we were able to see that despite the fact that 300 micromolar of agonist was present and the desensitized state should be stable, we see spontaneous open events again at a real high frequency, matching literature real good. So for now, it's my pleasure to thank you all for the attention, to thank our supporters and collaborators, and yeah, to say thank you. Ready for questions.
So I work at the Mayo Children's Hospital in Florence, and so our main focus are developmental and epileptic encephalopathies, which are a severe group of the neurodevelopmental disorders that are often associated with drug-intractable epilepsy. So we start from a clinical diagnosis of patients, and then we perform next generation sequencing studies to identify the variant, and most of them are of unknown significance. That means that they require a functional characterization to find if they have an impact in the patients. So what we do in the lab, we set up functional studies, mostly in vitro models, but also in vivo, with the aim of finding the pathophysiological mechanism that is the cause of this disease. And our main goal also is to find novel treatments for drug-resistant epilepsy. So we are going to acquire the patch liner, and we want to work with this patch clamp, because we think that we can accelerate the functional validation of VUS, but also we think that this instrument can facilitate the identification of treatments for these patients. So the poster that I'm going to have here is mainly about this channel that has been previously mentioned by the first speaker, STIM-63B, which is a such activated channel whose dysfunction is linked to a severe form of DEE. So we had a first project where we study the channel in a model of transfected cells, where we study the channel by stressing the cells with an hypotonic stress. And then I also show new data on patient-derived cells, where neurons have been studied by patch clamp and MEA recordings. And there is also a new topic, of course, where the patch clamp, the patch liner, would be used to characterize the gain or loss of function effect of variants in ion channel genes. And these are just some genes that we found mutant in the patient.
Hi, good evening, everybody. Today I'm going to talk about the BNLCL2 hepatocytes and the action of Cloto activator on these cells performed with the porta-patch mini device by Nanion. Cloto is a membrane insoluble protein, which is an anti-aging factor and also a modulator of oxidative stress and inflammation. In this experimental work, we have a compound which is a Cloto activator capable to induce Cloto production. And we tested this compound on BNCL2 hepatocytes, and we saw some results testing this compound with recordings performed with the porta-patch mini. Here we see two different conditions. We had the first condition where we tested Cloto activator at two different concentrations, 0.1x and 1x, and we saw an activation which was concentration dependent. And in the second condition, we also tested Cloto activator 1x and 5x, which was the highest concentration of this compound. In fact, we can see that with 0.1x, we have a minimal activation visible at around -100 and plus 100 millivolts. We have a greater activation and negative potential with the 1x, and we can see in both graphs where we tested Cloto activator in the first and the second one, that the current is about -1000 picoampere when potential is -100 millivolt. And the final concentration, the highest, was the 5x, where we had the highest activation of inward and outward currents, but was most visible at a negative potential, reaching a maximum current of -2000 picoampere at -100 millivolts. At the end, we tested the barium chloride tetraethylammonium solution, which was reducing inward currents in both conditions, and at positive potential did not completely block outward currents. So we thought that there could be a possible competition between barium and calcium ions on ketone channels which are active at this potential, such as TRP channels. Here we can see the trace recorded with the porta-patch mini, the original traces. We also saw that with the control in high calcium, which was a condition to have a better seal, we had higher outward currents, and so there will be also an interest of calcium in outward currents. Here we can see with the Cloto activator and the Cloto activator 1 and 5x that there is an increasing current concentration dependent. At the end, also we see barium chloride tetraethylammonium solution, which is closing inward currents and not totally closing outer currents. Here we have activation curves that are confirming what we have seen before. And so in the end, we also see here that with Cloto activator 1x and 5x, slope was steeper of both of the curves, indicating a faster activation kinetics of the channels involved. As I said before, we had the most evident effect between -50 and -100 millivolts. And this is making us think that there could be an involvement of potassium in rectifier channels. And here, this is our conclusion of these preliminary data. And another aspect that will be further investigated is the incomplete block observed with on outward currents by barium chloride and tetraethylammonium solution, which, as I said before, is supporting the hypothesis of ionic competition between barium and calcium. And thank you all for your attention. Thank to Nanion Technology for the invitation, and Professor Domenico Tricarico and my team. Thank you.
What I want to present on my poster is the work about the TMM175, so a lysosomal cation channel, in cooperation with many people from Milano, from Munich, also from Nanion, and from Bonn. And to remind you, so TMM175 was discovered as a potassium channel in 2015. Later on, there were studies that showed that TMM175 also conducts protons, but it's still an ongoing debate if protons or potassium are actually the physiologically important ions, and there are still people doubting that protons actually play a role, and this proton current could be just an artifact. What we are asking some questions that we want to answer in this project. So which ions are actually conducted by TMM175 under physiological conditions? Then is the activity of TMM175 pH dependent? And if so, which structural elements serve as pH sensors in TMM175? I only want to show one example experiment that I also have on the poster, and that if you want, we can discuss later. So we did several experiments, electrophysiological experiments, but also we did MT simulations. In this whole cell patch clamp recording, we started symmetrically with 140 millimolar of potassium and pH of 7.4, and then we exchanged the bar solution to a solution with pH 4.7, so very acidic. And what we see is that in blue and gray here, we see an increase in the outward current and an increase in the inward current. You also see here the corresponding ramp currents. And what is happening is that you see this increase in the conductance, and in red, you see a transient shift of the reversal potential towards the proton Nernst potential. And this already shows, okay, protons are actually conducted, but why is the reversal potential actually shifting back? So this is the question we wanted to answer, and I just want to provide you with the main findings, and then we can discuss, if you want, these findings and the experiments at my poster. So TMM175 conducts actually potassium and protons, and protons are 50,000 times better conducted than potassium under physiological lysosomal conditions, let's say. So pH 4.7 in the lysosomal lumen. And the TMM175 mediated proton conductance leads in patch clamp experiments to a fast dissipation of the pH gradient, even under high buffer conditions. TMM175 is gated by luminal, or in the whole cell patch clamp configuration, acidification, so on the extracellular side. And we found that histidine 57 on the luminal side of the protein acts as a pH sensor.